This project treats FAERS review as a screening problem rather than a diagnostic one. The source extract published by Kanchana on 1999 on Kaggle in 2024 contains 528,000 rows and 29 structured columns covering reports from 2015 through 25. From that extract, a balanced modeling sample of 50,000 rows was drawn, with 25,000 hospitalized cases and 25,000 non-hospitalized cases. The target variable is a binary indicator called each hospitalized. The question is not whether a model can diagnose harm. The question is whether a model can rank incoming reports so that reviewers open the highest risk ones first. That framing shapes every decision that follows, including the choice to balance the sample for comparison clarity, the choice to report ranking metrics alongside accuracy, and the refusal to describe any output as a clinical finding. What appears on this deck is a sorting tool built on reporting system data, and the narration will stay close to that boundary. FAERS review is a queue problem before it is a modeling problem. Reviewers at the Food and Drug Administration receive a continuous stream of post-market reports, and the operational question is which ones to open first. A fixed rule ages poorly because volume and hospitalization rate drift across quarters and years, and a purely manual triage process does not scale to the full 528,000 row corpus spanning 2015 through 2025. The business problem figure on this slide shows the mismatch between intake volume and review capacity. The goal is therefore not to replace a reviewer, but to order the queue so that the reports most likely to involve hospitalization surface near the top of the list. Prior pharmacovigilance work, including Demuchel in 1999 and Baton Evans in 2009, treats signal detection as a prioritization task rather than a classification verdict, and that precedent supports the framing used here. The deliverable is a ranker, not a rule, and the evaluation metrics on later slides reflect that distinction deliberately. The cleaned FAERS extract contains 528,000 rows and 29 columns, all structured tabular fields rather than free text narrative. Key variables include patient age in years, patient weight in kilograms, patient sex, country of report, number of drugs, number of reactions, year, quarter, and month. The binary target is hospitalized was derived from the seriousness flags attached to each report. Before modeling, 6 fields were excluded to prevent label leakage. Serious flags is fatal, life threat. Each one is either the outcome itself or a direct downstream consequence of hospitalization, so leaving any of them in the feature set would overstate model performance and produce a result that would not survive deployment. In the 50,000 row modeling sample, patient age was missing in 26.2% of rows. Patient weight (PGG) was missing in 69.7% of rows, and the dropped serious flags column carried 22,182 missing entries. Missing weight pushes that feature toward the lower end of the importance table, and it justified median imputation over row deletion. The modeling design is deliberately small so that the comparison stays legible. 2 models were trained on the same balanced 50,000 row sample with an 80-20 stratified split, producing 40,000 training rows and 10,000 test rows. Logistic regression served as the baseline, using standard scaling on numeric features and one-hot encoding on categoricals. Random forest served as the comparison, using ordinal encoding on categoricals, no feature scaling, 100 trees, and scikit-learn default hyperparameters, as described by Pedregosa and colleagues in 2011. Median imputation filled missing numeric values, and most frequent imputation filled missing categoricals, applied consistently across both pipelines. The primary evaluation metric is PR-AUC, because the operational task is ranking, not classification at a fixed threshold. Recall is reported alongside it because the screening use case penalizes missed hospitalizations more heavily than false alarms. ROC-AUC, precision, F1, and accuracy appear for completeness but are not the decision. Both models were fit in a single scikit-learn pipeline to keep pre-processing identical and the comparison clean. Exploratory analysis surfaced several patterns that shaped the feature set. Hospitalization rate rises monotonically across age groups from 0.464 in children aged 3 to 12 to 0.614 in seniors aged 66 to 80 to 0.674 in patients. Male reports hospitalized at a rate of 0.542 across 19,400 cases, compared with 0.487 for female reports across 26,066 cases. The drug count signal is strong and clean. Single drug reports hospitalized at 0.316, while polypharmacy cases with 6 or more drugs hospitalized at 0.602. Country-level rates vary substantially, from 0.405 in the United States to 0.882 in Slovakia, which is a warning that country is partly encoding reporting behavior rather than clinical bias. The logistic baseline reaches accuracy 0.6805, precision 0.6826, recall 0.6748, F1 0.678, ROC-AUC 0.7388, and PR-AUC 0.738 on the 10,000 row held out test set. That performance is above the 0.500 chance floor expected on a balanced sample, which means the structured features carry usable signal without any nonlinear modeling. The logistic coefficients are interpretable and largely consistent with the exploratory patterns shown on the previous slide, with age group, drug count, and country emerging as strong linear predictors. This model is the reference line. It is not the finished product, because the PR-AUC leaves meaningful room for a stronger ranker, and because the recall figure 0.675 is too low for a screening task where missed hospitalizations are the most costly error. The random forest improves every headline metric over the logistic baseline on the same 10,000 row test set. Accuracy rises to 0.6976, precision to 0.6857, F1 to 0.73, ROC 0.7. The most consequential change is recall, which climbs from 0.6748 to 0.7296. For a do not miss cases screening task, that 5-point gain is the operational reason to prefer the forest over the baseline. The mean decrease in purity importance table places country at 0.1376, number of reactions at 0.1 through 2, quarter at 0.1288, patient age at 0.1289, and number of drugs at 0.19. Country leading the list is a signal worth examining rather than celebrate. FAERS is a spontaneous reporting system, not a causal dataset, and the model inherits that boundary. Reports arrive from clinicians, patients, manufacturers, and regulators, and the rate at which a given country files reports reflects infrastructure and policy rather than drug biology alone. When country carries the highest feature importance in the random forest at 0.3076, that is evidence the model has learned reporting behavior in addition to any clinical signal. quarter and month, which together account for roughly 0.210 of importance, point the same direction. Hauben and Aronson in 2009, and Sarfman and colleagues in 2002 warn explicitly against reading FAERS signals as incidence or mechanism, and that warning applies here. The model ranks reports. It does not establish causation, and it does not estimate population-level hospitalization prevalence. The balanced sample was chosen for comparison. I recommend deploying the random forest as a queue sorting layer inside an existing human review workflow. Incoming reports would be scored in batch, sorted by predicted probability, and presented to reviewers in descending order. The model sorts. It does not close cases, flag specific drugs, or override reviewer judgment. Because country, quarter, and month carry substantial importance, a subgroup fairness audit should run before deployment, with particular attention to whether the ranker size temet I Kali Depri or ITISES reports from under-resourced reporting systems. I would retrain on a quarterly cadence using the most recent complete quarter as a holdout, which gives a rough temporal validation signal the current study does not provide. If I had additional time, I would tune the probability threshold against reviewer capacity, test a slimmer feature set that excludes the reporting behavior proxies, and stand up a monitoring dashboard tracking recall on a rolling basis. Those 3 items are the concrete next steps. 6 limitations constrain the conclusions. Reporting system bias is the most important, because FAERS overrepresents countries and periods with active reporting infrastructure. The balanced 50,000 row sample is not representative of population prevalence, and the figures on this deck should not be read as epidemiological rates. Imputation on 69.7% of weight values and 26.2% of age values introduces uncertainty that the model cannot express. Country, quarter, and month operate as proxies for reporting behavior rather than clinical biology. No temporal validation was performed, so generalization across quarters is assumed rather than demonstrated. The feature representation is single pass and does not exploit drug reaction co-occurrence structure. The repository contains the white paper, the deck, the speaker notes, the figures, the modeling outputs, and the notebook trail supporting every number cited. The open question for discussion is how a reviewer should use a ranker that orders reports well, while still reflecting reporting pattern signals.